for me to start my PhD with this exciting project, as well as the CAMDA organizers for giving me the opportunity to speak here and the gracious funding of the city of Vienna. To start off, why would I want to assemble metagenomes in general? So what are the gains compared to more read level based methods? So first off, and one of the most important things, I do not need any reference databases for assemblies, and that enables me to detect novel species. So if I'm able to assemble something I cannot identify before, it is probably a new species. And with these draft genomes I can obtain from de novo assemblies, they enable me a wide area of functional analysis of separate community members, so I can go down to the single number of bacterial species and see how they compare to each other. What I will give a short example of, I can estimate, for example, a replication activity of these single species in these samples, and many, many more. So what were my objectives with these challenge data set? I wanted to assemble and bin the urban metagenome sets, and to kind of have a control, I used mock communities of each single sample to optimize and to assess my methods. So to start off, how does binning in general work? So at the very top, these are single bacterial species. They are whole genomes shotgun sequenced and assembled. And these contigs are then separated by the tools I applied here with MetaBud, MaxBin, Concact. They apply nucleotide composition and differential abundance and coverage information. So what does that mean? They look at tetranucleotide frequencies in the case of MetaBud, which is the figure I took here from. And if abundance information, which means if I have a species with the abundance of 1 time and a species B with the abundance of 5 times more, I would also expect, roughly simplified, that the coverage of contigs originating from these species have an abundance of 1 or 5 times higher than the other, and thereby I can separate them. How do I assess qualities of bins I retrieve by these methods? What I applied for this is CheckM. This is using single copy marker genes. At first, the bins I recreate, which are the collection of contigs separated into different bins, that's what they call. They are compared against a reference tree with very universal single copy genes, and then a lineage specific marker gene set is selected from this placing in the reference genome to estimate the completeness and contamination. What does that mean? If I have, for example, 10 single copy gene markers as depicted here, if 7 of those are present, I estimate I'm roughly 70% complete. And if 2 of them are double, meaning that a single copy gene, as by the name, should only occur 1 time. If I have these in double triplicates or even the more, I have contamination originating from another strain or even from another bacterial species, telling me an estimate of how contaminated the bin is I retrieved from the binning process previously. The same single copy genes, not the exact same, but for the same kind of approach, I used as well for phylotyping with M4A2. So the bins I got in the end were assessed with single copy markers and used for phylotyping, and that I will show later on with some examples. So how does my workflow look like now? I start off with metagenomic samples. I assemble them with megahit, and these assemblies I obtain from, I bin with these 3 different binning tools, and thereby I get bins. These exact same samples here, already quality controlled I have to add, to create mock communities which are as similar to my original samples as possible to assess how well my methods work. I use Centrifuge, which is a CHEMA-based profiler, and take that taxonomic profile, take all the species level and lower taxonomic species, and see if I can find them in RefSec, which is a subset I have to add here as well, sample them in the exact same abundance I found them in the original file, and create my mock communities by them. With a lot of, as well, I add the unclassified reads, because unclassified does not mean that it's not bacterial, it's just that I don't know it. And thereby I have a mock community which has the same roughly bacterial profile than my original samples, but as I took it now from RefSec, I do know now exactly what kind of input reads I had in there, and thereby I can assess how well my methods work, and they are processed the exact same way in the other samples as well. Just in short overview, you've heard that already multiple times. I used Sacramento and Boston data. I did not use the New York data because the sequencing depth for assembly was a bit too low. So that did not really work out. The touchscreen samples of Boston as well as from Sacramento are presented in a bit more detail with the examples I take later. A short overview, what I looked into the data, okay, how many bacterial reads to begin with do I even have, or how does it roughly look? So this is a output from centrifuge. How many, how this is Sacramento, so you can see that the sequencing depth is rather even, around 20 million reads. And I have about 10 to 25% of bacterial reads, a varying amount of eukaryotic, and roughly half or a bit more than that of unclassified reads. Same goes for the Boston data. Here you can now see that the sequencing depth is of a much, much higher range, which was already mentioned previously as well as there are samples with a huge majority of eukaryotic reads, up to sequencing of 40 million to less than 1 million. So that is much more difficult now to assemble as well with some examples. I will show that later on to give you a rough idea what the input data looked like. Now for the mock communities, from the same thing as I mentioned already previously, I take them with the same abundance obtained from the taxonomic profiles, add the unclassified reads, simulate them with a Phred score of 20, do the same thing with the triple abundance to kind of increase the abundance, see how much that improves my binning with unclassified reads. I take them with the same abundance without any unclassified, kind of a pure mock in a sense, to see, okay, what does that have an effect on? So I was playing around with these, added the quality and multiple mores, but these are the ones I will show later on. So these are now the assemblies I created out of these samples as well as mock communities. So the green one here is the Sacramento ticket machines and the respective mock communities from sample 1B. B are the ticket machines, and the Boston ones to the bottom. And what you can see here, so these are single sample assemblies as well as here. And these, Sacramento had all around 20 million, and this respective sample number 5, this from Boston as well, had about 20 million reads. But you can see that my assembly size is much, much higher in the Sacramento one than the Boston. And why is that? When I looked at the original from the sheet, I saw that in Boston I had a much less sequence DNA, input DNA for sequencing. So what I hypothesized here, especially as well as the pool, that even though I was sequencing to a much better sequencing depth, as I had too little DNA input, the rare species were not sequenced that well, and they still had a much overrepresentation of like the most abundant ones, thereby not making it possible for me to properly assemble the low abundant species as well. But if I now pool, and I pooled per surface type here, I have to add, if I pool all the ticket machines from Sacramento, as well as these are 6 samples with each 20 million, and these are 9 samples with that wide range, I again come to a comparable assembly size, which does tell me to the first hand, these samples are comparable, as when I add them together, I add up the rare species, and are thereby again able to assemble also less abundant ones, so they're comparable in essence, and that sequencing depth alone is not enough for detecting these ones, so I also have to take care of previous ones as DNA input and further on. If I now have these assemblies, this is a short overview of the Sacramento ticket machine bins I can create. I cut off at 50% completeness, estimated by CheckM as mentioned previously. And the ones I want to look here is the completeness on the X and the contamination in the Y-axis. And this bin, for example, here in blue, which is obtained from Metabot, I will show a more detailed analysis later, giving me 90%, less than 2% contamination, and a length of 2.9 megabase pairs, which I'm going to be quite happy about later. So this is the number of bins, but you can see that there's a wide range of completeness, as well as some of them have very high contamination, which can originate from a large number of multiple strains from the same species, which make it very complicated for the assemblers and as well as the binners to really separate them again. So a high species heterogeneity, as well as very similar ones, do have a major impact in these, which is a point I will add on later as well. No, do you want? There you are. This is a representation of all the bins from all separate sample types. So from Boston Grip, Boston Pole, Boston Seed Bag, Touchscreen, and so on. And what you can see here is I excluded all bins with 0 completeness, and I took the average completeness to kind of estimate, okay, where does they place from, and as a kind of indication how many reads that I had classified as bacterial, which I could then assess with CheckM as this is only going for bacteria mostly. What you can see here is that even though with the Boston samples, which are here more to the right, I had not that much input, but I got a very few, as the size is the number of bins I got, but very complete bins. As in contrast to the Sacramento samples, I got a lot of bins, but they were in average, there were a lot of those which were less complete in general. So why is that? I can, again, as I added before, when I have a very low diversity, as the rare species were not properly sequenced out of whatever issues it is, this makes it very easy for me to assemble the most abundant ones because I have less disturbing factors, and thereby I can obtain very complete and very clean bins. As in contrast, if I have the real diversity, including a proper sequencing depth of the low abundant ones, this does affect my assemblies negatively. So the standard assembly parameters where you go for kind of the best contig sizes and the best N50 do not very nicely apply to metagenomic data as such, because just when I have a very nice assembly studs in very complete bins, it might mean that I just got the most abundant ones and my low abundant were far too little sequenced. So it does not represent my true diversity of the sample. What you can see here as well, that in terms of assembly statistics from the single samples, and these N50 values and so on, which are very often used, were actually consistently worse in the Sacramento samples because the diversity was much higher. I kept it, and that basically disturbed my most high quality bins. But nevertheless, I was able to get very high quality bins, and with those I can then do further analysis. And what I'm going to show here is an example. This is a bin from the Boston set I mentioned here, and with the tool IREP, I can now, with a bin which has at least 75% and less than 2% contamination, estimate the replication rate of that species in my sample. How do we do that? IREP predicts the origin and the terminus of replication, as when a bacteria replicates, it starts with the origin of replication. There's a bidirectional replication. So I have a higher coverage at the origin of replication and at the terminus. And if I then sort my contigs and I have a predicted terminus and origin of replication, I can then estimate by the skew of this coverage if there was a replication or not. I have to stress out here that this is not directly a death or alive ratio, because what it does tell me that previously, before I sequenced, there was replication. But if I look at the touchscreen, this can as well be just deposited bacteria which died immediately off. If I want to know more information, I would probably need to, for example, I would need to do time series. But to bear that in mind, this is not exactly death or life, but it still does give me a very nice indication if there was active replication or not. So one has to be aware of how one takes his results. And as you can again see here, the completeness was near complete with no contamination detected, very few contigs per megabase pairs, and Propionibacterium acnes, as mentioned previously as well, is a very commonly known skin bacterium, and the published length of the bacterium is again as well 2.5 megabase pairs. So the contamination estimate of 99.34% seems to be very plausible, and the IREP value of 1.37 in this case here means that with an IREP value of 2, it would roughly mean that about every bacteria in my, from this species was replicating. So with 1.37, about a third of those seem to have been replicated prior to sampling. Same example with a bacteria from the Sacramento 1B sample here, Coccura rhizophila, reported to be found in the soil, but as well again as also on the skin. So it does make sense that I find it on touchscreens. Here I have a bit less completeness and a bit more contamination. As you can as well see that my contigs are a bit more. The length is 0.2 megabase pairs longer than the one reported in literature. But again, I can estimate an RREP of 1.83, so it seems to be quite nicely replicating. I have an average coverage of 9.42. So, and as well, again, 29 of 31 markers from amphora do tell me on the species level this is Cochlear rhizophila, which I've been here. So in conclusion, it is possible to do genome-centric analysis over metagenomic datasets. Near complete draft genomes with minimal contamination are able to be achieved. And not only sequencing depth, but also the DNA input do impact the detection of rare species. And from my observations here, an interesting thing that I want to do, try it very shortly as well, is it might be an idea to subsample very diverse data sets to improve the quality of the high abundant ones, as I kind of miss out the low abundant ones and can improve the high abundant ones. And with the achievement of these high complete bins, I then can do further really detailed function analysis on these single species, and that opens a wide area of very, very interesting research questions. And with that, I want to thank you for your attention, and I'm happy if there's any questions to take them.
Great, any questions? Great talk.
A great talk. So I was wondering if you have compared this to kind of reference-based approaches, and then just taking your bins and seeing how much is novel you detect, and then if bin corresponds to any novel, to the new or existing organisms. And then I would imagine that would be very different for the bacteria, where it's much harder to assemble versus like eukaryotes or the viruses.
I have to say that there are bins which are there, which are not really classified, so they're kind of classified as bacteria, not any further. I did not have the opportunity to really investigate them further, but this is again this is one of the very first next steps I want to take, because these are in fact the ones which are the most interesting then. They might represent novel species, they might represent some kind of erroneous assemblies, so that needs quite a thorough analysis, but in the end these are the most fruitful ones. So they are there in the data set. They are about, I had some from the pooled samples, I have that in the single samples, they were usually not that detectable, but from the pooled ones I had with 80% estimated completeness, so that is already quite neat in this regard, and less than 20% contamination. But I have to say I cannot honestly answer yet, as the analysis was not done thoroughly enough.
But what is your expectation, like eukaryotes versus like bacteria? Is it true to say that to assemble eukaryotes is much simpler, just because there is so much less gene transfer going on, for example, which is like makes your graph much simpler to assemble?
If I would have a pure eukaryotic sample, probably, but in this very mixed.
But is it feasible to extract the eukaryotes and then assemble them separately?
It is, well, if you map against the kind of reference, you definitely can extract them and assemble by themselves. I have to say that I did not try this out.
Okay, thank you.
You're welcome. Thank you.
One quick follow-up question on what else you can get from the assemblies. Did you look at all for some of the things like biosynthetic gene clusters or other functional elements that might be pulled out from the assemblies?
We did not look at that, but when I really got when I proved that these bins I have are really clean, and I got that working, which I'm pretty much at the moment right now, this is definitely the next step to go for.
Great. Yeah, and also we've been running metaspades, and we could send you all those files. It'd be interesting to compare the assemblies.
Already talked with David.
Yes. And also these assemblies will be part, when they all get finished, the goal is they're also trying to release them probably as part of the CAMDA challenge maybe for next year as well. It's not just reads, but assemblies, but we can talk about that later. But okay, no other questions? Thank you.